So let's start talking about codon charts and how to translate, from our perspective, these actual physical letters and these codons into amino acids. So first thing you'll notice here is I have drawn a strand of nucleotides up here. I'd like you to tell me if this here is DNA or RNA. Go ahead and pause if you need to. All right, you should have said this is DNA, right? This is DNA. This is DNA because it has T's instead of U's, right? So this is DNA, and this would be the non-coding strand or the antisense strand. Again, we have some complementary mRNA that's going to be made down here, and we'll assume this is mature and everything. This is our mRNA, and you'll notice it's complementary. T binds with A binds with U, C binds with G, and so on and so forth. You guys can make sure that I did all that correctly if you'd like. Hopefully I did. But the point is, this is that mRNA that's being made. Now, if we want to figure out what amino acids are being made, it is a three letters in our nucleic acid language. Correspond with one amino acid, right? So three letters in our nucleic acid correspond with one single amino acid. So this AUG is going to code for a specific amino acid. So the way to read these charts is to look for this first letter here, right? First letter of this first, again, what are these three letters called here? You should have said codon, right? This is a codon chart. After all, these first three letters are going to code, or the first letter of this first codon here is A. So go ahead and find first letter over here and find A, and we've got it. Second letter, find it up top here and find out which box those two meet in, which is right here. And then we look for the third letter, G, and we follow that along to find out what amino acid we're making. It turns out we're making methionine, which is normal because this is in red over here, if you'll notice. And the reason for that is this is typically our start. Amino acids are going to start with an AUG signal. Okay, I'd like you guys to go ahead and pause and go ahead and take the rest of this and go ahead and try and decipher it and tell me what's going to go in these other circles over here. For GGC, I'm assuming you're back now. Hopefully now's your chance to pause it if you didn't pause it. Should have gotten glycine, right? G, first letter, second letter G, and C, GLY, right? That should give you this amino acid right here. Next one, AUU, first letter A, find that over here. Second letter U, same box as methionine. And then third letter U right here. You can find that right here in this box, and that gives you isoleucine, ILE. And then finally, if you did the last one, UAG, that's going to lead you to, and I already had that one filled out, but first letter U, second letter A, third letter G right here. That is a stop codon that says, hey, stop, and let's go ahead and terminate this sequence. And then you have your polypeptide or protein right over here. Right? Ready to go and be folded and do whatever its purpose is. Whatever this protein is supposed to do. The last thing I want to kind of touch on is how translation is a little bit different in prokaryotic organisms. As you'll notice left here, we have a prokaryote. On the right, we have a eukaryote. Let's go ahead and review this process that occurs in eukaryotic organisms. We have DNA that we have pre-mRNA transcribed from. Of course, if you'll notice, these introns here get cut out, right? You don't notice these dark red maroon portions in this mature mRNA down here. So we have introns removed, we have a GTP cap, and we have a poly A tail added to it, and it's ready to go out here and be translated by ribosomes over here, which are translating it, right? Again, many ribosomes are translating this, and you have many times, so a lot of protein is being made, right? Eventually, this thing will get eaten up and destroyed. right? Now, in the prokaryotic organisms, the difference is you don't have processing. You don't have intron removal or any of that stuff. When you have an mRNA made in a prokaryote, it's immediately, right? The ribosome is almost right here next to the DNA because it's not contained. Remember, it's not contained in a nucleus. So it doesn't really have the time to prep this mRNA. This thing's already out in the prokaryotic cytoplasm immediately. So once that mRNA is hot and fresh right off of that DNA, it starts getting translated into proteins without any processing whatsoever. So because there's no nucleus in prokaryotic organisms, right? There is no processing, right? There's no processing of the mRNA. The ribosomes just kind of translate what they can before that mRNA is eventually broken down. So that's pretty much it for transcription and translation. If you need help with that, let me know. But I'm going to go ahead and leave it at that for topic 6.3 and 6.4. Thank you for listening.